Next up we have Fiona Radcliff. Fiona is from the University of Auckland, did her undergraduate at University of Tasmania, New South Wales for a PhD, Helicobacter pylori antigens, Institut Pasteur, and then made her way back through Australia to New Zealand to work in John Fraser's laboratory. And you're going to talk today about an mRNA vaccine for Staph aureus. Thank you, Fiona.
Thank you, Gary. You missed out a little diversion into biotech as well in Auckland with Genesis. But, I can see you've done your homework. And I apologise for my slides. Unfortunately, the laptop here remodelled them a little bit, but tried to fix them. So a change of tack here to Staph aureus, which is a bacterial pathogen our group's been working on for some time. And I'm sure most of you don't need a reminder about what Staph aureus does. We've all encountered Staph at some point in our lives, often pretty early on. It's an opportunistic pathogen which can cause skin and soft tissue infections, or alternatively, for example, if you go in for surgery, then it may cause complications there and very serious infections. It has been listed by the World Health Organization as a high priority pathogen for development of new antimicrobials because of what's been described recently as the silent pandemic of antimicrobial resistance. So we definitely need better treatments for this organism. And also Staph aureus is actually a pathogen that is present in higher rates in New Zealand than many other developed countries. And it's also important for New Zealand because it's an important veterinary pathogen. So it causes mastitis in dairy cattle. It would be one of the major causes there. So obviously the current treatment for Staph aureus is antibiotics, but along with many other people, we think that a vaccine would be a better alternative to prevent this infection, or at least limit the infection with Staph. So a lot of people have been trying to work on vaccines for Staph aureus. Several of them have gone into clinical trials. To date, unfortunately, with the same negative outcomes. One of the issues, which I think was touched on by one of the speakers earlier, I think perhaps talking about latent viral infections would be Staph aureus, in common with those, has an issue with lack of immunologic correlates of protection. So we don't actually really know, or don't have a good correlate of protective immunity for Staph. So that's one of the challenges, I guess, in developing a vaccine. What we do know, however, is that innate immunity is very important for conquering, or at least controlling, a Staph infection. So our vaccine is based on 3 virulence factors. They're all from a family called the staphylococcal superantigen-like proteins. So these virulence factors are important in immune evasion, and they do target the various facets of innate immunity. So we've chosen 3 of these. I should add that these are common to all Staph aureus strains, so clearly very important. So the ones, the pointer is working there. So the ones that we've selected are SSL3. So this is a protein that will completely block signalling through toll-like receptor 2, which is an important innate activation pathway for Staph aureus. SSL7, which binds both IgA and also complement C5 and causes a pentameric complex. So it prevents IgA from binding to its receptor, and it also prevents C5 from cleaving into C5, most importantly. So that's a potent chemotaxin for neutrophils. And SSL11 is the other one that's in our vaccine. So this is a protein that binds to carbohydrates, primarily on myeloid cells and particularly neutrophils. And this protein gains entry into the neutrophils, and by mechanisms that we don't fully understand, basically stops them in their tracks. So they will not migrate to the site of an infection anymore. So these 3 very critical virulence factors that we've chosen for our vaccine. We've done a little bit of work in mice. This was done some years ago now by a PhD student in our lab, showing that if you knock all of the SSLs out of Staph aureus and then measure survival of Staph aureus in human blood, the survival of the bacteria is significantly diminished. However, if you restore the genes for SSLs 3, 7 and 11 back into the Staph, then you will get improved survival of bacteria again. And similarly in mice, without the SSLs, Staph doesn't actually survive quite so well. So moving on to our vaccine strategy, these 3 proteins are actually very well characterised, so we've been able to mutate them so that they're no longer active through targeted mutation, and we've combined them into a fusion protein, so have the 3 proteins that could be expressed together in E. coli. We've done studies in mice to show that mice that are immunised with these 3 proteins as a fusion stimulates very strong protective immunity against an intraperitoneal Staph aureus challenge. That's a disseminated infection with staph. And what actually surprised us, and we still don't fully have the answer to this, is that actually if you immunise the mice with these 3 individual proteins together, you don't actually get protection. So there's something important about fusing these 3 proteins together to help our vaccine work. What piqued our interest in the RNA field was that ultimately we've been doing this work using a mouse protein, but we want to obviously tackle human disease primarily, and to do this we think that we will need at least 6 different constructs to cover the main Staph aureus clonal types that are prevalent globally. So we were very fortunate to have the opportunity to join Duccio's team for the R3 platform, or program, sorry, not platform, to then check whether our Staph aureus vaccine approach, a protein approach, could be translated into an mRNA vaccine. I thought I'd just take you through a few of the steps that we went through in this process. So I've just got a Western blot here showing our recombinant bacterial protein. The first version of our mRNA vaccine, actually, we're perhaps a bit naive about this. Glycosylation was mentioned earlier this morning. We found that our original transcript actually was glycosylated when it was produced in mammalian cells. So we made a second version of it, which is called the deglycosylated PSSL. And we found that this bacterial protein was produced in reasonable levels in a mammalian cell line. We have a collaborator who's got a lot of skill in codon optimization, so to improve protein output. And so we had 2 versions of our sequence. So the second one, we got much higher production of protein. And I should add, we used the BioNTech 3' or 5' 3' UTRs. And this was produced as a secreted protein. And we also looked at reactogenicity of this sequence. So these are modified RNAs, so we've got pseudouridines in them. So you can see that there's low reactogenicity in a THP1 cell line. We did also do a little bit of work exploring our original mRNA transcript. So this was work that was done with a different collaborator. So what we were comparing was a transcript that hadn't undergone codon optimization. We had another one that had been was codon optimized with the goal of reducing its reactogenicity, because obviously native RNA does actually have issues with reactogenicity in mammalian cells, and that can reduce the production of protein. And then we compared that same sequence with the incorporation of pseudouridines. So THP1 cells obviously are bristling with pathogen recognition receptors, so they would only make protein when the pseudouridines are incorporated, whereas 293 T cells are not at all fussy in that way. And more importantly, we also found that, unfortunately, the intention of the codon optimization was to reduce reactogenicity, but didn't actually have any impact on our sequence in this particular sequence. So that particular codon optimization strategy was not suitable for us, so we had to go with pseudouridines, basically. So we then moved on with our chosen vaccine and tested that in mice. So we're looking at production of antibodies and also looking at cellular responses, and we compared that to our protein vaccine, our lead candidate, as a benchmark. And we found that we needed to do you needed to use 3 doses of our mRNA vaccine to get equivalent or higher antibody responses as compared to a protein vaccine. And we also found we got very strong cellular responses, particularly Th1 responses, so mostly gamma interferon producing CD4 and CD8 T cells in both the spleen and draining lymph node, which I guess is what you would expect from a lipid nanoparticle vaccine. We also were interested in whether we could get neutralising antibody responses from this vaccine to the different components within it. So we found that we got strong or equivalent neutralisation of SSL3 with the RNA vaccine, and also very good neutralisation of SSL7 compared to the protein vaccine. So we're really we're very excited about these results leading into a challenge study, because they looked to be as good or superior to the results that we got with our protein vaccine. And unfortunately, we found that when we went and challenged mice, sure enough, the mice that received our protein vaccine actually did really well against challenge. This is in 2 independent studies, but unfortunately, the RNA vaccine failed to protect these animals. So obviously, that was a disappointing outcome. We think that one of the ways that we can get around this issue is obviously this LNP formulation that we've been using has perhaps got some things missing that we need to, we need some extra signals in our vaccine so that we can get protective immunity to staph. So I'll just go to touch briefly on some of the things we've started doing to work towards this in the New Zealand platform, and that's looking at alternative LNP formulations with Gavin's team. So to prepare ourselves for this work, we've had a look at a few different LNP formulations, some novel ones that have been made, and we've used the Firefly reporter system and in vivo imaging just to have a look at where these LNPs are going, and the length of time that they're being expressing expressing protein. So the benchmark we're using is ALC 0315, which obviously gives good expression and good distribution of protein in mice. And the lead lipid that we found out of this screen was lipid 1C, which has got equivalent performance to our benchmark. We also, because we use a 3-dose immunisation schedule, we needed to have access to LNPs that were stable out to 6 to 8 weeks after they've been produced. So we did recently get some results that suggest that both of these LNP formulations are active out to about, they both have equivalent performance out to about 10 days of detectable signal. It's 0, 4 and 8 weeks after they've been produced, so that's a good result. And even more recently, we've also looked at some Firefly luciferase that has been made here in New Zealand. We were initially using commercially produced mRNA, but because of the tyrannies of distance, it's not ideal to have to keep purchasing RNA in from overseas. So we now have some great mRNA that Rebecca and the team have made here that performs just as well as the commercial material. So I guess where we're at the moment with our vaccine is trying to think about what I guess I've just called the X factor here is for our staph mRNA vaccine. So we think that perhaps incorporating, for example, a toll-like receptor agonist into LNPs may be a solution to build on the humoral and cellular responses that we already get with our lipid nanoparticle vaccine, but perhaps widen the scope a bit so that we get protective immunity to staph. There's also perhaps the potential to move from using a secreted protein to perhaps trying transmembrane expressed proteins as well. Word through the grapevine is that that's actually a strategy that might improve responses to bacterial vaccines. So there's a few different things we can try here. And lastly, just to wrap up, I've got a lot of people to thank. I've got in the lab there, we've got Nilu and Mel who've done a lot of the hands-on work. Reese, who's very disappointed that he couldn't make it today, is our molecular biologist. Obviously, we've been working with Gavin and Sarah and Rebecca and Caitlin here with the New Zealand-based work. And of course, we got a lot of our work done under the auspices of the R3 platform with Duccio and other collaborators there. And I'll leave it there. Thank you.